Okay.
Hello, hi everyone. Good day, good morning, good afternoon. We hope all of you are happy and healthy and also safe. And we wish everywhere on this planet Earth to be safe and in peace. Welcome to the webinar of today, 24th of February 2022. Sebastian, myself, both proudly from TU Delft. We are excited to have this gentleman here, very close to my office recently. Um, are pleased and delighted to host you all who joined this webinar, and also our distinguished guest, Professor Julia de Rezende from Heriot-Watt University. Actually, we're part of Sebastian's heart still is. So the former colleague of Sebastian. Julia is a geomicrobiologist who integrates uh, anaerobic microbiology, molecular microbiology, bioinformatics, biogeochemistry, and modeling to understand and analyze microbial ecopsychology in the subsurface and engineered systems. Julia's work targets the delivery of science based microbial monitoring and control solutions looking at activity rates and persistent microorganisms that can cause longer term damage to infrastructure operations and the environment. Julia did her PhD at the Center for Geo-microbiology in Aarhus University in Denmark, followed by a postdoc at Newcastle University, and has been at Heriot-Watt University since the establishment and opening of the Lee Center in 2016. So it's a pleasure, true pleasure and honor to host you, Julia, today. Thank you very much for graciously accepting our invitation during the academic year. Uh, and to the audience, please note Julia's lecture would last for about half an hour, 35 minutes, followed by questions. Like always, please do type your questions in the chat box, and Sebastian will take them like always. Without any further ado, Julia, the stage is all yours.
Thanks very much, Haji, and thanks, Sebastian, for the invitation. As we were saying earlier, it's really a pleasure. It's a fantastic series, and it's great to be part of it. Yeah, great to talk to a different audience as well and hear different questions, hopefully. My name is Julia Rezende, and I'll be talking about some of our recent work on microbiome monitoring and control in engineered systems. So just a couple of examples from studies that we have done on reservoir microbiology and the lessons that we can take from there into the new adventures of geoenergy, so to say. So let me see if this is working. Okay, yeah, right. So first of all, I actually want to thank Nicole Dopfel, who gave a talk here at the seminar series a few weeks ago, and it was really great. And if you haven't seen it, I recommend just going back on the list and finding it, because she was talking about life in the subsurface, and that makes my introduction a lot simpler. So I'm not going to spend a lot of time here, but if you missed it, and haven't had a chance to see it yet, then just as a quick reminder, there is a lot of life beneath our feet on planet Earth, and underground life is actually a lot of life on our planet. So it's estimated that there are about 10 to the 30 microbes on our planet. That's such a huge number that we can't quite get what does that actually really mean. But on top of that, 10 to the 29 of those microbes actually live in the oceanic and the terrestrial subsurface in our planet. So it's really a lot, and it's quite impressive when you think about how much attention is given to that huge number of microbes versus how much attention is given to the minority of the microbes in the rest of our planet. So especially the ones that are related to the human body, the ones that cause disease. So pathogens are really just a minority. And all of these other incredible diversity and incredible number of microbes, they have a variety of functions in the functioning of our planet, and the ones in the subsurface are no exception. these so-called interterrestrials, they form a microbial community that is really resilient and really pervasive, and finding, ways of surviving in every different type of environment in our planet, including in the crust of our planet. And just because they are down deep, that doesn't mean they are separated and isolated from the rest of our world. And in fact, they are in integral players in our global cycles. They contribute to climate regulation, for example. And one of the many functions they have is that they are able to degrade this very deeply buried carbon. And with that, they release minerals, they release nutrients from these rocks that, with fluid flow in the subsurface, can come up again, and like I said, interact with our surface environments as well. So it's really impressive, because it's something that we haven't even realized that there would be any life at all in the subsurface until relatively recent. So now there's so many questions, so many questions we can ask about it. How does that life work? It's a very different type of environment, and the conditions are different. Even the time scales are also different. So there are lots of open questions there. And also we wonder what happens when we interfere with that environment. Every time we drill, every time we introduce fluids, every time we remove things from the subsurface, we're changing that environment. So how does the microbes that are already there, how they respond to that? Also, when we end up introducing more microbes into those subsurface environments. So there are lots of questions to be asked, and today I'm going to focus on a specific type of subsurface environment, which are oil reservoirs. And I find them fascinating, deep biosphere environments, for many reasons. They're quite different from the general subsurface. They're like an oasis in terms of the type and the amount of organic carbon that you can find. And again, so many questions that you can ask. We wonder whether microorganisms have been in reservoirs since their formation, and if not, then when did they get there? How did they get there? How they survive all this time? But we also wonder what the consequence of the activity, both over time. So, for example, they are involved in degradation of hydrocarbons in petroleum, and in the long term, the formation of heavy oil deposits. But also in present day, they associate with production of a variety of undesirable substances like H2S, hydrogen sulfide, or polymers, organic acids, and so on. They can also be involved in microbially influenced corrosion, which affects infrastructure, for example, the well integrity, not only in oil and gas, but in other industries as well. So that's a very costly microbial problem. And they can also disrupt flow by forming biofilms that clog pores and so on. So they also they can also have good roles, and in Nicole's talk she also talks about some of those. But here I'm gonna focus on only a couple of examples. In this lecture, what I wanted to communicate is just 3 take-home messages, 3 lessons from recent studies of ours on the microbiology of oil reservoirs. The first one that I wanted to take home is that subsurface microbes, they are natural survivor specialists. They are really tough. They can survive conditions that you wouldn't expect, and they're still there. Also, in a way related to the first one, unwanted microbes can be difficult to kill. So sometimes we benefit from the fact that they are really tough. Sometimes we don't. So when you're trying to actually get rid of them, you can face a lot of trouble. And the third one is that monitoring these microbial communities, monitoring microbial dynamics, is key for a long-term management of your engineered system. So to give an example of the first point, I want to go back to a study that I've done with colleagues in the University of Calgary in Alberta, in Canada, where we looked at Athabasca oil sands deposits, which are the result of millions of years of microbial degradation of light oil, forming these large deposits of heavily or severely biodegraded oils. That are the tar sands or the oil sands. And that's quite a large area in Alberta, and it's not pretty. if you've seen photos of this before, so it's to be able to use these oil deposits, you have to remove all this overburden, and it's a environmentally quite taxing process, and it can be controversial, and it's complicated, right? And one question that we had in mind was, could we actually find a better way of using these oil sands without removing them from the reservoirs? So could we, for example, use these oil sands as feedstock for the microbes that are already there, and see whether we can stimulate them to produce methane, and then we use the methane only and leave the oil sands where they are, if we're able to also keep the CO2 underground. So that would be perhaps a better way of using oil sands. So we set out to ask that question, and we know that this could be a possibility because there are several mechanisms from which methane can be produced from degradation of crude oil, and we've known that from degradation of various types of oils. But the question was whether oil sands specifically, being so heavily degraded, whether that could be a feedstock that can be used for methane production. And if so, we wanted to know at what rates, and which microbes would be involved in this process. So if we find out who the ones that really are key to deliver this, can we then find ways of stimulating them in an industrial way? We also wanted to know if which oil compounds we preferentially degraded in the process, and whether this can be then a greener energy production from oil sands. So this is some work that we published back in 2020, but actually started a long time before that. So the samples for this experiment were collected back in 2005 by one of my long-term collaborators, Casey Hubert, also a PhD co-advisor, and Thomas Oldenburg, Milo van Fustik, and colleagues. And they got some formation water, they got some oil sands, and made these microcosms where the oil sands were the only carbon source for the microbes. And the microbes came only from the oil sands or from the formation water itself. There was nothing else added besides just some inorganic nutrients and a buffer. And then these bottles, these microcosms, were then left incubated with nothing else added for 3,050 days. And that's not a typo. I'm really talking about 8 years in a bottle with nothing else added to it. So what we saw was that there was a generation of methane from oil sands. It was possible, but it was very, very slow. Long story short, it was not a viable solution for green extraction of oil sands. It's not something we would recommend pursuing. There's a paper where we talk more about it. And the main reason is because the rates were very, very slow. So here we compared the rates of that experiment to other experiments in literature where methane is being produced from other types of oils. And you see it can be orders of magnitude slower than in other cases. In this graph here, the black bars corresponds to experiments where the microbes come from the same source as a carbon source. So you see it can be much better if you start with much lighter oils than the oil sands. So that was one of the conclusions, but the other conclusion was also that the native microbes in these oil sands are still able to live on very recalcitrant organic matter, so very hard to eat carbon sources, and that's also quite significant. So we saw that even if we left them just with that very difficult carbon source to eat, and left them in a bottle, almost forgot about them, you still have a whole diversity of microbes that are able to grow in those bottles, being sustained by very limited organic matter. And that was very interesting. And especially when after that we decided to, this is during those 8 years, we were measuring methane production, and we saw very, very little being produced over time. That's these open circles here, when they had only oil sands as a carbon source. But at the end of those 8 years, we opened some of these bottles and we took out some of the liquid, some of the microbes, and we put them into a new medium, a fresh medium, with benzoate as a carbon source, so much easier carbon source. And after a lag phase, we saw a very rapid production of methane. So remember, these guys had been in the oil sands for millions of years. Then we put them in a bottle and left them there for 8 years. Not very much more to eat there. And after that, we took them out and put them into new medium and added benzoate. And it took a while for them to wake up in that sense. But once they did, the production of methane was very rapid. It was actually comparable to when we looked at river sediments with a very active microbial population and lots of organic carbon sources. So the production rate of methane increased 110-fold once we introduced a more easily degradable carbon source. And the take-home message from this is that if we introduce fresh organic matter into the subsurface, that can quickly stimulate microbial activity. So going back to that original question, what happens to the subsurface when we change it, when we introduce new nutrients and new conditions? It can take a while for microbes to pick up, but once they do, they can actually act quite rapidly and can make a difference. In this case here, we were interested in methane production, but sometimes we're not interested in the products of microbial activity. So when that's the case, when you have unwanted microbes, you might be more interested in stopping the activity rather than stimulating them. And that brings me then to the second example that I wanted to discuss today. For that, I'll be focusing on reservoir souring, which is one of the major issues caused by microbes in oil and gas industry. Reservoir souring, if you're not familiar with the term, it refers to the production of hydrogen sulfide, especially by sulfate-reducing microorganisms, and this is especially a problem in offshore industry, where introduction of seawater with lots of sulfate stimulates these sulfate-reducing microbes and then produce hydrogen sulfide. So I've been using this scheme from my colleague and friend Sven Lammer for quite a few years. I hope he doesn't mind that I'm using it again here. I'm still too lazy to make my own, but it's just to give it a little bit of an overview of what is souring. So during secondary oil reservoir oil recovery, there's a lot of seawater is injected into the reservoir, and with that, a lot of sulfate. That is great for sulfate reducers who may come with the seawater is injected or may already be in the reservoir. And there's perfect conditions for them to then use the sulfate to produce hydrogen sulfide, which then accumulates, and that's souring, and can lead to corrosion, stress cracking, and health and safety issues, and increases production costs and so on. So it's not something that we want to have in the system. One way of trying to control that is by adding biocide to hopefully kill everything, or at least the sulfate reducers. And biocide, as a treatment to mitigate microbial problems, is quite a common strategy that's used a lot. But we know that eventually it fails. You can't just apply biocide once and then go home, problem solved. And the question that we want to ask is why is it that it fails? Why? How does the microbial community react to this treatment, and what can we do to make it better? So for that, I believe we need a better understanding of how complex microbial community react to biocide treatment. But often the research about this is done on a single microbial species in the lab, and sometimes even microbes that have nothing to do with the system in question. So we wanted to change that. We wanted to look at in more detail how a complex microbial communities react to biocide treatment. And we know that biocide use is strictly regulated, and it should be. So in our case, we want to provide science-based evidence to inform regulators and industry on the best ways of using biocides in terms of concentration, in terms of long-term strategies, so that we can minimize the amounts needed, both for cost but also for environmental safety, but at the same time maximize the long-term effect of these treatments. So for that, we did one of the experiments that we did, the one that we'll be talking about here. We used microcosm bottles again, This is a paper that we recently published in AM. And there we tested glutaldehyde, and another aldehyde releasing biocide. And we made these microcosms, and we added a single dose of biocide at the beginning of the experiments, and then monitored the whole system for 12 months. In this case, we used a souring microbial community that we had enriched from river sediments as a proxy for reservoirs. And we added sulfate, we added some easily accessible carbon source like acetate, propionate, and butyrate, and we incubated all of that at 25 degrees. So in some of the bottles, we had no biocide treatment at all, no souring control at all. And in other bottles, we had 100 ppm of glutaldehyde or 750 ppm of glutaldehyde, or the same with the second biocide, ARB. We had 100 ppm or 750 ppm. And for each of these treatments, we had a killed control as well, which is autoclave, so treated at high temperature before the start of the experiment. And that should kill most microbes. Should. So what we saw in terms of souring control, as you'd expect, in the bottles where we had no biocide at all, there was no souring control, of course. So there was a very quick sulfide production as soon as the experiment started, and within 2 months, essentially, they had run out of substrates, they run out of organic substrates, and had produced quite a lot of sulfide. And then after 6 months of the experiment, we decided to add more nutrients and see if that would, if the microbes will pick it up again. And yes, of course. So after that, they produced more sulfide until they ran out again. So this is what happened when we had no microbial control of any sort, just lots of sulfide being produced as expected. Now, when we added biocide, every single scenario gave us something different. So where we added 100 ppm of the ARB biocide, or the aldehyde releasing biocide, we saw there was no sulfide production after 7 days. So it controlled souring for a week. But then soon after that, everything looked exactly as the no biocide treatment. So these are the yellow lines on this plot here. And you see it looks very similar to the no biocide profile. So that wasn't a great souring control strategy. When we added glutaldehyde at 100 ppm, we saw a slightly better control. So again, we saw no sulfide in the first 7 days, but then it's increased a little bit more slowly. But by day 63, again, we have more or less the same pattern as we've seen for no biocide. So a short-term souring control. For the high concentrations, at 750 ppm, the ARB biocide controlled souring for the duration of the experiment. So remember, I've added only a single dose at the beginning of the experiment, and we saw no sulfide at all for the subsequent 12 months. That's pretty good. For glutaldehyde, however, at the same concentration, we saw effective souring control. But then for every one of the 3 bottles that we had, the replicate bottles, we started seeing sulfides being detected again. So one of them, we had sulfide being detected after 97 days, then the next one after 166 days, and the third one after 187 days. So there was souring control in this case for a longer period of time, but still there was failure after some point. It can still be considered somewhat successful because we're talking about a single injection of biocide in the beginning, and also the total amount of sulfide that was produced was about half of what it had been produced in other cases. So that's not too bad. But we wanted to look a little bit more into what happens after biocide fails. Just as a quick note, the killed controls, all killed controls that had biocide, with the exception of one, they remained pretty killed. So there was no sulfide being produced in these controls. But for the control that we had only treated with temperature, we still saw some sarin or some sulfide being produced. That's a slightly separate story. That's because of autoclave resistant sulfur reducers in the sediment. That's, we can talk about that later, but in any case, if we look at the autoclave and biocides treated, we can see that that controlled sarin quite well, as expected. Now, this can be a complicated graph. I'm just going to go back here. Throughout this whole experiment, we took samples from each of these bottles, each of these systems at every time point. So each one of these time points here, we collected sample for DNA extraction, and we did DNA sequencing to characterize the microbial community, to see who is present in these in these bottles during the experiment, before and after a biocide fails, for example. And the analysis that you can see here essentially compares the overall profile or the fingerprint of this microbial communities. And when we see two data points that are close to each other, that means the microbial community is quite similar between these two samples. And the further apart they are, then the more different the microbial community are from each other. So if we go navigating this slowly, you can see here that in this part of the plot, we see all the samples that were associated to treatments where there was effective sarin control, or that they were just inactive. There's nothing going on. So this essentially is the region of the plot where we see good sarin control, not much going on. That's how the microbial community. That's where the microbial community fits. Essentially, that's where you find those points. Over here, a little bit below, we see the type of microbial community where there is a lot of sulfide being produced. There's no sarin control. So all the black dots here are from the untreated, and the yellow dots are from the low ARB one that behaved exactly the same as the no biocide treatment. So you see there's a difference in the microbial community, and they start to cluster separately. Over here in green, these are the samples from the treatment where we had 100 ppm of glutaraldehyde. So we had a short-term sarin control, but the presence of the biocide caused a change in the microbial community. So if we look at just the H2S, it's not very different, but when we look at the microbial community, we see that the presence of the biocide caused some change in the microbes. So the microbes that were responsible for sarin in this case are different than, for example, the microbes that are responsible for sarin in this case here. Now the blue dots is the one that I find quite interesting. So these are from the bottles that had 750 ppm of glutaraldehyde. And here we saw longer sarin control, but we also saw some significant community change. So if we go back, talking about these bottles here. So remember that there was sarin control for a while, but then they started failing one by one. So after 97, 166, and 187 days. See. Now, on day 63, this point here, you can see that there is no sulfide detected in any of these three bottles of the blue lines, of the 750 ppm of glutaraldehyde. So no sulfide being detected, but when we look at the microbial community, we see these 3 points, the A, B, and C. And interestingly, they correspond to the bottles that were about to fail. So this point C here, it's the bottles that showed sulfide 1 month later. This one showed sulfide 3.5 months later, and this point showed sulfide 4 months later. So essentially, you start seeing they start moving. The microbial community pattern started moving from the fingerprint of the group that looked like sarin control was effective, and slowly moving over to this side where we saw no more sarin control post biocide failure. And that's essentially because the types of microbes that were present there were changing over time, and you start getting this signature of microbes that were associated with sarin under the influence of biocide. And this is one way of showing that the monitoring of your system is key. So again, we don't need to rely just on seeing sulfide already, because that's already too late. If we're able to monitor the system and see how the microbial community is changing by using molecular ecology techniques, we might be able to get some early indications of upcoming biocide failure. So it helps us understand our system and get these early warnings before the problem is already too late. Another way of looking into this was when we looked specifically at the sulfur reducers in the system. So we saw that the presence of glutaraldehyde led to an increase, a relative increase of sulfur reducers that are harder to kill. So spore-forming sulfur reducers are known for being more resistant in adverse conditions. So we can see that the usual suspects, or the usual sulfate reducers that would be present, or that were present in the untreated bottles, they were essentially gone from the bottles with glutaraldehyde, and instead we had an enrichment, which are the darker green colors, of sulfate reducers that are able to produce spores, and therefore they're harder to kill. So this is analogous to the situation that when you have, for example, an infection and you have a course of antibiotics, you are told to have it for, the right dosage for a certain length of time, and your GP will tell you to stick to the recommended time. Don't stop your course of antibiotics before the recommended time, because otherwise you're just killing the easy to kill, and then you leaving yourself with the harder to kill ones. And that's a similar case that you can have in your engineered systems as well. And this also shows that it's important to understand which sulfur reducers are present in the system, because that can help troubleshoot and that can help tailor mitigation strategies for a longer term positive effect. Because otherwise, you might be getting a more difficult problem over time. You're killing the easy ones, and you're enriching the harder to kill in every single step of treatment, and that can be a lot more difficult. So once again, monitoring is key for the success of microbial control in engineered systems. So we've also done other types of work looking at biocide theta and the effect of microbial communities in sand packs and flow-through systems. So I'm not going to get into detail here. And if my recently graduated PhD student, Zhang, is in the audience, you'll pain him to see his entire work in a single slide. So I apologize for that, Zhang. But it's just to say that we also looked at systems with flow, so not only static systems, and we looked at the effect of glutaraldehyde on souring. We saw that again, it can be only temporary, and we also saw that the ineffective treatments can cause the problematic microbes to go deeper into the system. So essentially, by adding ineffective amounts of biocide, you might kill the ones that are closest to the inlet, to your injection point, and that's essentially favors a nutrient abundance to go deeper into the reservoir, and that favors microbes deeper in the reservoir to then survive. So your treatment might not actually be reaching microorganisms at the effective concentrations if they moved further away, essentially. So it's really essential to understand microbial distribution and the transport in subsurface, but also it's very important to understand the heterogeneities of the microbial community, who is where, as well as the physical-chemical environment. So in conclusion, again, I'm going back to those three take-home messages, but in more detail, especially because I see that I'm over time. I wanted to highlight that microbes, as you'd expect, can cause a variety of issues in engineered systems. And that with subsurface engineering, you might change the physical chemical conditions of that environment, and this has a potential to stimulate microbial activity. That's as I give the example with the methane producers in oil sands. And this could be because of introduction of nutrients and microorganisms. That could be because of increased leaching of minerals and organic matter from subsurface rocks. And when that's a problem and you want to treat them with biocides, well, there might not often be a simple and effective solution. And it can really cause operational environmental risks. So it's something to look into and see how can we make this treatment strategy better for a longer term. And I would say you should really pay attention at the long-term buildup of biogenic gas. That could be an issue for the infrastructure, that can be an environmental issue, that could cause contamination, for example, when you're talking about hydrogen, underground hydrogen storage. And even if you think as far as decommissioning, so every operation that we're starting now, we're going to have to end it at some point. And if you have a slow activity of microbes that, at some point is going to cause a problem, then we should be monitoring early enough to know at that point what we need to do, or at least to avoid getting to a point that it becomes very expensive and very difficult to deal with. And I advocate that we need to better understand the dynamics of the microbes, the microbiome in these engineered environments, especially the complexity of microbial communities and the interactions of the environment. But I think the question for me for this decade really is how to best integrate microbiology and geoenergy. So there are lots of fundamental questions in microbiology. We're never going to run out of questions to ask. And I think it's to get a good shortcut, so to say, I think we need to talk between disciplines and see how can we communicate better, how can we ask the right questions for lab experiments, and how can we use the immense amount of data that we get from for example, DNA sequencing analysis, and bring that back into application in the field, for example, via models. And that's one way that we're trying to do this at the Lyle Center at Reservoir. So yes, I think I'll stop here. And of course, I've showed a little bit of our work, but a lot of people are involved in this. So there are many people that I should thank. This work was mainly in these two recent papers, if you're interested in a bit more. Thank you very much for your attention. I'm happy to answer any questions if there are any.
Thank you very much, Julia. It was very, very informative talk and presentation. I see already many posted questions, and we try to also there ourselves also to bother you with some questions as time allows. So Sebastian, please.
Yes, thank you very much, Julia, for a great presentation from my side as well. Yeah, as Heidi said, a number of questions have already appeared. So I start with one from Marty Bone, and she's asked, the pressure and temperature is very different in your bottles from the experiment. Can you still see this as a representative experiment? What would happen in the subsurface?
Yes, that's a good question. So the pressure is definitely a factor, and we don't have a lot of experiments with very high pressures. We do see that temperature has a much higher impact than pressure. There is a limit, both in terms of pressure and temperature. We see that with pressure, we see a reduction of the, it affects the physiology, but it doesn't affect so much the type of microbes that we find that are active in sarin, for example. But they might be adapted to higher pressures as well, and it's a bit difficult for us to do the lab experiments unless we have taken the right samples, they're preserved in the right way, and conserved all the way from sampling to bring into the lab, to running in the lab without releasing the pressure. So this has been done, but it's a little bit more limited what we have learned from it. But still, in terms of temperature, 25 degrees, I know that is quite low, but we see a lot of the mixing zone in the lower temperature reservoirs. A lot of the microbial activity will happen closer to the injector well, where the temperatures are going to be lower because of the introduction of seawater. So this is still a relevant temperature because it might be one where most of the microbial activity will be happening in certain reservoirs.
Okay, thank you. And you sort of touched upon bringing the samples into the lab and making sure that they are sort of preserved properly. And Marty had a follow-up question along those lines. Says, I'm not an expert, or I've read somewhere that it's very difficult to cultivate microbial communities obtained from the subsurface in the laboratory. Could you comment on that?
It can be. So that's why we use molecular techniques where we don't necessarily depend on cultivation. So we're able to sequence DNA, for example, from subsurface environments and get a snapshot of microbes that are living there, and sometimes even reconstruct the entire genomes without actually cultivating them in the lab. There's only so much that we learn with this, so we can generate a lot of hypotheses of what they are doing in the subsurface. But then if we're able to do lab experiments, then we can test those hypotheses. We can do physiology experiments, and both are being done. It's a lot of work is being put in multiple groups around the world in subsurface microbiology for that. But we also see that we still have lots of similarities with microbes that we can find in analogous environments as well. So, for example, the sulfur reducers that we find in reservoirs are often the same sulfur reducer that we find in different environments in marine sediments, for example. Even so, going back to the temperature, we find different types of microbes at different temperatures, but again, they're not necessarily exclusive to the reservoirs. We can find in different environments as well. Even though the spore-forming sulfur reducers, in fact, we can find some that are happy to live at or work at 50 degrees, even 70 degrees, and we find them in cold sediments, sometimes just being dormant and waiting for somebody to pick them up and put them in a higher temperature. So there are differences, but we can use analogous samples to work with these microbes and learn more about how they function.
Thank you. And you spoke about the sort of variability of microbes, and Wilhelmine van Rooyen says, thank you for the interesting presentation. And her question again is along those lines: are there different kinds of microbes present in different locations? I think the answer is yes. There from you, but is there a way to predict the types present in a reservoir and to predict the risks of those unwanted microbes, or do you need to drill and see what's in the subsurface and then act accordingly?
Yes, that's a very good question. So there is what we call like well, at least we're seeing that there is a kind of a core microbiome. So the microbes are typically found in oil reservoirs and subsurface in multiple places. But that doesn't mean that they're always going to be exactly the same. So also understanding the conditions and what's not just the temperature, but what kind of nutrients you find. So you have some that are present in many, and some that are specific. It's a bit of a balance of both. So I think you had a second question. Your question, and maybe I only answered part of it.
Is there a way to predict the risks of any unwanted microbes being present?
Prediction is always a bit complicated, I'd say. I guess you could have educated guesses, but I would suggest that it's relatively easy enough to characterize the microbial community, at least as a first snapshot, by taking good samples from this whatever new environment you're at, and doing 16S RNA gene sequencing, for example, as primary characterization. So I would say we don't need to predict without taking a good sample and looking at it. At least if you're able to take one sample. I would advocate that the more samples you take of good quality and the more frequent monitoring you have, then the better you can capture the dynamics of the system. So when they start changing and some of them start growing faster than others and taking over, so we can't do a full prediction of a successful prediction, I would say, but you can have an educated guess based on what we have seen before, the types of microbes that we would expect to see in the different conditions.
You spoke about the monitoring, and two questions that are somewhat related. One is from Mr. JVIJ. You mentioned monitoring the microbes. Is it only possible to take samples from a reservoir? Are there also physical indications that can be observed using existing methods? And Yang Wang sort of asked more specifically, can you comment on how microbes impact things like viscosity of the fluid or permeability? So what do we need to do to monitor, and what's the impact of the microbes on subsurface flow?
That's great questions as well. So we can indirectly look at microbe activity by, for example, looking at isotopes, isotope signatures and fractionations, for example, or production of certain biogenic compounds like hydrogen sulfide. You're able to tell whether that comes from microbes or not. So we could indirectly look at that, but by also looking at with DNA sequencing or physiological experiments, seeing which microbes are actually doing that, that can also help. If you want to control that, can also help and see, okay, these type of microbes, maybe they work better in these conditions. So can we change the conditions to unfavor them, for example, or the other way around, if that's actually something that you want? So what can we do to stimulate these type of microbes? We can even play microbes against each other, so to say. So one way of controlling souring is by injecting nitrate. And the basis for that is that it stimulates nitrate reducers who then can compete with sulfur reducers and reduce souring production. So understanding the system, seeing who else is there, can help us find like smart solutions to deal with microbes. Yes, I think, sorry, maybe I picked up on a tangent here. So yes, you can look at it essentially to indirectly look, see microbial activity. But I think we are at a stage that we can use multiple tools in our toolbox of monitoring, and the question is, why not? So shouldn't we combine all these?
And in terms of the petrophysical properties that Yuan asked about, is it mostly that permeability is affected, or is it more an issue with the fluid properties themselves, that fluids become more viscous or degrade? What are the change composition? What is the key challenge?
Yeah, I think that's difficult for me to say. So I think you can see changing in viscosity, changing permeability. We can see the production of gases. There's so much. I think maybe I'm not the best person to tell you what are the best factors to look at. It's microbial growth or microbial biofilm formation. I think we might not even know very much in detail, because the different biofilms might have different effects as well in terms of the characteristics of the type of biofilm. So yeah, I think I might need to pass on that one.
Okay, no problem at all. We have a question. Take two quick questions. We have one from Hazrizal, and he says, "Nice and great presentation, Julia. My question is there any specific reason to choose GLUT versus RB versus THPS?"
Yeah, so thanks, Arizo. So glutaraldehyde is one of the most common, usually biocides. That's why we chose that one. The second biocide, the AB one, is one that was provided by a collaborator. So we were interested in comparing. It's a relatively similar type of biocide because as glutaraldehyde, because it's also an aldehyde releasing one. But it shows us that for regulation, if you just say the maximum we can add is 200 ppm, no matter what, then it can have very different effects if you have a different type of biocide, even when the mode of action is relatively similar. So understanding the like how to choose concentrations and to minimize it, but it's still getting something effective. It might not be like a single number for everything. I think it's important to look at specific effects of certain biocides. THPS would also be a good candidate. It's very commonly used as well, but for this study we didn't. We had to limit it, so we kept only 2.
Okay, great. Thank you very much. I think the last question that Marty had was sort of similar to some of the previous ones around the impact on reservoir behavior, and if there's something else. You talked about oil and gas versus CO2 storage, for example. Don't know if there's anything you want to just make a final comment on microbes in other, not just oil and gas, but say in CO2 storage sites and hydrogen storage sites. But this may as well be just another full talk, I guess.
Yeah, no, it would be. I think my final comment would be that there's a lot of experience on microbial issues in oil and gas, and I think we don't have to reinvent the wheel as we move on to other ways of using the subsurface and other geoenergy operations applications. So I think it's important to learn from what we have seen in the past decades in terms of microbiology of reservoirs, and seeing, okay, how can we apply this knowledge now to the new ways of operating, and monitoring is one of them. So in my view, start looking at your system before you have a problem. It would make it a lot easier to deal with the problem when it comes, and it most likely will come at some point. But also there are many open questions about how microbes deal with hydrogen being injected in the subsurface. So as I said in the beginning, it is going to be a big change of the environment. And Nicole in her talk, she talked a lot about it as well. So hydrogen is a fantastic nutrient for microbes, a substrate for microbes. So it is very much expected that if they're there, that they will use it. But there are many questions about how quickly they would, and if the very high partial pressure of hydrogen that's quite unnatural in a way, whether that would be something that would stimulate the microbes or would actually inhibit them. So it's really, there are lots of unknowns, but I think the only known thing is that it's worth looking at those questions now, while we're in the beginning, than just wait to see the problem and then start dealing with it.
Great, thank you much. Thank you on that note. Hand over to Hardy again for some final comments. Thank you, Julia, for a great talk, making the time to give the presentation, and to our audience for excellent questions. Thank you.
Thank you very much.
Thanks all. I'll take the chance quickly to announce our next week's speaker will be Professor Gregory Shapiro. We are going to move all the way from Scotland, where Julia is, now to Brazil. Gregory would be our first Brazilian speaker from Brazil to speak. We have had many Brazilians in the channel. Perhaps Julia, you yourself are from Brazil as well. So, but we are so excited. Gregory is a dear colleague in mathematics department in Federal University of Juiz de Fora. Did I pronounce well?
Yeah, okay. Awesome.
Yeah, it's great. So we are gonna go there in Brazil. Well, virtually we are gonna go there. I wish we could really go there, but we are not being able to. So stay with us. Gregory will speak about a non-Newtonian flow of foam in porous media. So until next week, stay happy, healthy, and we sincerely wish everybody on this beautiful planet be on peace and happy and healthy as well. Stay with us until next week. We see you all, and thanks a lot, Julia. It was our pleasure to host you here.
Thank you. The pleasure is mine. Thank you very much.
Bye-bye, everyone.
Bye, everyone.